I don't know why it does this.
The fat.
Oh, hold on. Let's make sure we're playing. Maybe I could share the entire screen. Sure. I don't know. Sure. Let's see. Oh, interesting. It only works with YouTube. I didn't know that. And I guess let's try the entire screen today. Maybe I should do that, but then I lose the ability to share. I don't know. Let's try the entire screen, I guess. Also share system audio. I'm not sure. I don't know if you guys can see then. Sure. I don't know.
So don't use these two terms interchangeably. They are not the same exact thing, but they are related. One causes the other.
Come on, buddy.
And this is the fact. So don't use these two terms interchangeably. They are not the same exact thing, but they are related. One causes the other. Now, let's discuss the mechanism by which HIV actually invades the white blood cells of our body. And let's begin by examining the structure of an HIV agent. So this is what HIV looks like. We basically have this membrane shown in pink, and on the membrane we have these embedded glycoproteins. Now we have different types of glycoproteins. The two glycoproteins we're going to focus on in this lecture is a glycoprotein looks like. We basically have this membrane shown in pink, and on the membrane we have these embedded glycoproteins. Now we have different types of glycoproteins. The two glycoproteins we're going to focus on in this lecture is a glycoprotein called GP120 and glycoprotein called GP41. So GP120 is positioned on top of GP41. Now GP120 is actually used to bind onto receptors on the target cell's membrane, and GP41 is used to stimulate the process of fusion of the membrane of the virus and the membrane of the target cell. Now inside this membrane we have the protein capsid shown in brown, and inside that protein capsid we have the contents of that virus. We have the genetic information, the RNA shown in red, and we have two very important types of protein enzymes, one known as integrase. Let's suppose that's the blue one, that's the green one, and the other one is known as reverse transcriptase. Let's suppose that is our blue one, and we'll discuss what their function is in just a moment. So basically, the GP120 glycoprotein of HIV binds to a specific glycoprotein receptor found on the membrane of immune cells. And this special type of glycoprotein is called CD4 glycoprotein. So HIV contains a glycoprotein, the GP120, that can bind onto the CD4 protein of immune cells, our white blood cells. Now, recall from our discussion on white blood cells, and specifically on T lymphocytes, we said that one of the specialized types of T lymphocytes that contains a CD4 glycoprotein are the helper T cells of our adaptive, our acquired immune system. So recall that helper T cells are CD4 positive cells. And what that means is these HIV agents will be able to bind and will be able to attack these helper T cells. So these are the helper T cells that HIV actually attacks. So let's take a look at the following diagram. We have the HIV agent that uses the GP120 glycoprotein to bind onto the CD4 glycoprotein of this healthy helper T cell. Now, inside the helper T cell, we have these different types of organelles. So we only show the nucleus and we show our endoplasmic reticulum. This is the rough endoplasmic reticulum that synthesizes our protein. And we have the genome, the DNA of that helper T cell inside the nucleus. Now, once this binding process takes place, what happens next is the GP41 glycoprotein, the other glycoprotein attached to the GP120 and to the membrane of our virus, initiates or stimulates the cell-mediated endocytotic process. This is the process by which the membrane of the virus fuses with the membrane of the cell. And that essentially dumps all the viral contents into the cytoplasm of our cell, as shown in this diagram. So we have the fusion of the membrane in pink of the virus and the membrane in blue of that helper T cell. So this is what happens. We have the blue section and we have the fused pink section. And the protein capsid along with the contents are now found inside the cytoplasm of our cell. So what happens next? Well, HIV is a type of virus known as a retrovirus. Now, what exactly is a retrovirus? Well, a retrovirus is a virus that contains RNA molecules as well as a special enzyme that catalyzes the formation of viral DNA from that viral RNA. So that's why we have reverse transcriptase, because it synthesizes our nucleotides in the opposite direction to what is normally seen in nature. Normally, we synthesize DNA to RNA, but in this case, it goes from RNA to DNA in the opposite process. So HIV is a retrovirus, which means it has an enzyme called reverse transcriptase that can form viral DNA from the viral RNA. So these blue molecules, once inside the cytoplasm, attach to the red RNA molecules, and they begin to synthesize the viral DNA. Now, once the viral DNA is synthesized, it moves into the nucleus of that helper T cell, and an enzyme known as integrase, shown in green, basically binds onto that DNA of that cell. It clips it, it opens it at a certain position, and it inserts, it incorporates that viral DNA into that original genome, original DNA of the helper T cell. And now, this is an infected helper T cell. So notice in this case, this was purely brown, but now a small piece of that viral DNA shown in red has been integrated with that genome of our helper T cell. Now that we have incorporated the viral DNA into our DNA of the cell, when that DNA is used to synthesize mRNA, we're going to synthesize viral mRNA to produce viral proteins. And in this manner, we transform our helper T cell into a factory that produces viral proteins and viral RNA molecules that ultimately causes the cell to lyse, and when it lyses, when it breaks open, it releases many HIV agents that go on to essentially infect other helper T cells of our body. And in this method, what HIV does is it essentially destroys our population of helper T cells in our body, and that weakens our immune system and leads to the acquired immune deficiency syndrome. And because we have the
plasma, we don't have any plasma cells. We won't have any memory B cells. The immune system's cooked. Literally.
Acquired immune deficiency syndrome, we have a very weak immune system. Any type of infection that enters our body can essentially kill us off, and that includes the common cold. So it's not the actual HIV, that virus, that kills us, but it's what it does that ultimately kills us. It's the fact that we create a very weak, or the virus creates a very weak immune system, and any infection whatsoever can essentially kill us off. Now, the next question is, what's the big deal about these helper T cells? cells. Why is it that when these helper T cells are destroyed, our immune system becomes so weak? So we have many different types of white blood cells in our body, and helper T cells are only one of these different types of white blood cells. So what exactly makes the helper T cells so special? Well, it turns out that helper T cells are the cells that oversee all the different types of processes that take place inside our immune system. And they essentially allow that immune system to function effectively and efficiently. And although they do not attack pathogens directly, what they do is they bind onto T lymphocytes and they induce the differentiation of these T lymphocytes into other specialized white blood cells, such as cytotoxic T cells, that are important in fighting off infections and infected cells. They also bind onto B lymphocytes, and that induces the differentiation of these B cells into plasma cells that are needed to produce antibodies floating in our blood, and they also induce differentiation into memory B cells that store the antibodies in case we are ever reinfected with that same pathogen. Now, these helper T cells also release special chemicals called cytokines. And these cytokines not only induce white blood cell differentiation, but they also stimulate the cells of our innate immune system. These are our macrophages, neutrophils, basophils, and other white blood cells of our innate immune system. So we see that by destroying the population of our helper T cells, what HIV actually does is it severely weakens not only our adaptive, our acquired immune system, but also our innate immune system. And as a result, most individuals infected with HIV die not from the virus itself, but from other infections that infect our body, such as the common cold, pneumonia, the flu, or even cancer. And because our body cannot actually defend itself because our immune system is weakened, that is why we actually die. So we die because of our body's inability to mount the proper immune response as a result of the acquired immune deficiency syndrome, or AIDS. Now, the final thing I'd like to briefly discuss is the stages that the person goes through when they are infected with HIV. So there are 3 stages. We have the early stage, we have the middle stage, and we also have the late stage, which is the stage when the person is said to have AIDS. So early stage lasts about 2 weeks, and this is when the person actually feels sick. This is when the person experiences flu-like symptoms. For example, they have a high fever, they have different types of body aches, they have headaches, and this is when inside the blood there's a high level of these viral agents, the HIV agents. So basically the individual experiences flu-like symptoms, fever, aches, and so forth, and has a high level of virulence in the body. Now, the middle stage is the stage when the person doesn't actually experience any symptoms. So you have no way of actually knowing whether or not you are carrying the virus. The only way that you can know is if you go get tested. And that's because this is the stage when the body begins to produce antibodies. So we have antibodies floating in our blood that are specific to this virus. And these antibodies can be used to detect that virus in our body. So middle stage can last from several months to several years. So no visible symptoms, low levels of that virus, we have the presence of these antibodies, and we have a very slow but very continual decline in the population of our helper T cells. So during the middle stage, the HIV continually but slowly begins to kill off these helper T cells. And the final stage, the late stage, this is when we actually are diagnosed with acquired immune deficiency syndrome. This is when our HIV begins to kill off the helper T cells at a very high rate. And the population of helper T cells drops to a very low amount. And what that means is this is when our innate and acquired immune systems are weakened to the point where if an infection takes place, such as the common cold, flu, pneumonia, we essentially will die because of our body's inability to create, to mount some kind of defensive response. And this is when the person can also develop cancer, and that's because our immune system loses its ability to fight off cancerous cells. Because if we can't produce cytotoxic T cells, and because if we can't release cytokines that influence natural killer cells, we don't have any cells that can destroy our cancer cells of the body, and that's why we ultimately develop cancer when we are at the late stage, when we have the acquired immune deficiency syndrome. The overall goal of our immune system is to protect the healthy cells of our body from invading pathogens, such as bacterial cells or viruses. It's to generate and maintain a state of homeostasis. Now, as with many things in life, our immune system is not a perfect system, and sometimes it does make mistakes. Sometimes the immune system incorrectly labels an otherwise non-harmful foreign substance that enters our body as being harmful, as pathogenic, and this elicits, it initiates a defensive response called an allergic reaction. And that causing agent that caused that allergic reaction in the first place is known as an allergen. Now, different people are allergic to different things. So allergens differ from one individual to another. People are generally allergic to certain foods or drugs or certain things found in our environment. For example, people can be allergic to pollen found in grass or flowers. They can be allergic to certain foods, such as peanuts, or certain drugs, such as penicillin. Now, let's discuss the mechanism by which an allergic reaction takes place, and let's focus on some of the, or one of the most common types of allergic reactions known as hay fever. Hay fever is not an actual fever in the sense that it doesn't actually increase the core temperature. Instead, hay fever is simply the name for the allergic reaction that takes place when grass pollen actually enters the air passageways of our lungs. So one very common allergic reaction is hay fever, which is the way our immune system actually responds to grass pollen. When the allergic individual inhales pollen, that pollen, which is essentially a microscopic particle, can begin to release antigens that our body sees as pathogenic. So let's take a look at the following diagram to illustrate how our mechanism is actually carried out. So let's suppose this is the air passageway, the bronchiole of our lungs. These are the epithelial cells as shown, and these are the microscopic pollen particles. Now, as the pollen particle moves, it can release these tiny particles, even smaller particles, that our body labels as pathogenic. So these are basically seen as pathogenic antigens by our body. So these tiny antigens can diffuse across the epithelial cells and into the nearby tissue where we have different types of white blood cells. So B lymphocytes produce plasma cells, and these plasma cells can recognize these antigens and begin to produce the corresponding antibody that binds to this antigen. And the most common type, the predominant antibody involved in allergic reactions is immunoglobulin E. So immunoglobulin E is produced by the plasma cells. Now, what these immunoglobulin E antibodies do is they move onto the cell membrane of special white blood cells found in the nearby tissue known as mast cells. So these mast cells contain these receptors that can bind to the constant region of our antibody as shown in the diagram. Now, once the binding takes place, now these antigens that are released by the pollen particles can move on and bind onto the variable portion on our antibody as shown in this diagram in section 5. And once this binding takes place, these mast cells in the cytoplasm contain these granules, these tiny vesicles that carry special chemicals, special immune chemicals, such as histamine. And once the antigen binds onto the antibody found on the membrane of these mast cells release these vesicles, these granules, and in turn release the histamine and other immune chemicals. Now, what the histamine and these other chemicals do is they initiate an inflammation response, and that dilates the blood vessels that carry blood to this area, to this infected area. So we say infected because this otherwise non-harmful foreign substance was labeled as pathogenic by our immune system, and so this technically is an infected area to that particular individual because the immune system of that individual treats the pollen antigens as pathogenic. So more blood flows into our infected area as a result of this vasodilation process, the increase in diameter of those blood vessels. That brings more blood as well as more white blood cells. Now, what histamine also does is it increases the permeability of the nearby capillaries to water, and that makes those capillaries leaky. And that's exactly why people that experience hay fever have a red nose as well as a leaky nose, because the capillaries become more permeable to water and we have more blood flowing to this area. And that's exactly why it appears more red than usual, because of the effect that histamine has on our body. So once again, tiny pollen, microscopic pollen particles enter the air passageways of our body, our nasal canal, our trachea, the bronchi, and the bronchioles. And they release these tiny allergens we call antigens that flow via these epithelial cells and into our plasma cells. And the plasma cells see these antigens as pathogenic and begin producing the corresponding complementary immunoglobulin E, our antibodies. Now, these antibodies then bind onto our mast cells, onto these special receptors, and now these antigens can go on and bind onto these antibodies antibodies at the variable portion, and when the binding takes place, these release histamine and other chemicals that basically dilate the blood vessels, and that brings more blood to that area, causing redness. It also increases the leakiness of our capillaries, which can lead to edema, the process of swelling. It can also create what we call a runny nose. Now, this type of allergic reaction is not that dangerous, but sometimes individuals are extremely allergic to certain types of things, such as peanuts or penicillin, and that can be very lethal if not treated immediately by a medical professional. Immune surveillance is the idea that certain white blood cells of our immune system are continually and constantly keeping a watchful eye for any abnormal cells that might develop in our body, and these abnormal cells might develop as a result of some type of pathogenic infection, or they might develop as a result of some type of carcinogens. So when healthy cells are exposed to carcinogens, such as chemical agents, pathogenic agents, or radiation, for example, UV or X-ray radiation, the healthy cells can essentially become cancer cells. Now, how exactly does a healthy cell become a cancer cell? Well, these carcinogens essentially affect the DNA molecules within our healthy cells. And when we develop a mutation in our DNA molecule, we change the sequence of nucleotides on that DNA. And that ultimately codes for different proteins, and that can influence the functionality of the cell. Now, we develop a cancer cell when our abnormal cell essentially loses its ability to control the way that it divides. And that's exactly why cancer cells divide rapidly and uncontrollably and eventually form large visible masses we call tumors. And because these tumors are so large, they can affect the way that our body functions, and they can ultimately kill that organism, kill that individual. The question is, not everybody actually develops cancer in their lifetime. And because on a daily basis, we produce anywhere from several to 1,000 of these different cancer cells, how exactly does our immune surveillance system, our immune system in general, actually detect these cancer cells and destroy these cancer cells? Well, since these cancer cells have a slightly different sequence of nucleotides on their DNA, they will produce slightly different proteins. And these different proteins that end up on the membrane of these cancer cells can be read by certain white blood cells as being foreign antigens. And when these white blood cells locate these foreign antigens found on the cancer cells, they can basically mark or label these cancer cells for destruction. So let's actually discuss what the different types of cells are in our immune system that basically are used to control and destroy these cancer cells. So even though the immune system as a whole functions to detect and destroy these cancer cells, two particularly important types of white blood cells involved in the process of destroying infected and cancer cells are natural killer cells, as well as cytotoxic T cells, also known as killer T cells. And these are two different types of white blood cells. So let's begin with the natural killer cells. So natural killer cells are specialized lymphocytes, and they have granules that carry digestive enzymes, as we'll see in just a moment. And these granules, these vesicles, play an important role in destroying those cancer cells. Now, let's recall the important points. So the majority of the white blood cells in our immune system are able to differentiate normal cells from abnormal cells, as well as communicate with one another as a result of these special protein complexes found on the immune cells and our normal cells of our body, known as the major histocompatibility complex, MHC. And we have several classes. For example, we have MHC class 1 and we have MHC class 2. Class 1 is used to differentiate abnormal cells from healthy cells, and class 2 is used to actually communicate between the white blood cells. So virtually all cells in our body, all white blood cells require these main the white blood cells. So cells and class 2 is used, and we have MHC class 2. Class 1 is used to differentiate abnormal cells from healthy cells, and class 2 is used to actually communicate between the white blood cells. So virtually all cells in our body, all white blood cells require these major histocompatibility complexes to actually interact with one another. These white blood cells. So virtually all cells in our body, all white blood cells require these major histocompatibility complexes to actually interact with one another and detect these abnormal cells. But the very unique thing about natural killer cells, which by the way are part of the innate immunity of our immune system, the very unique thing about these natural killer cells is that they don't actually need the major histocompatibility complex to actually find what our abnormal cell actually is, unlike most other cells of our body, including cytotoxic T cells, the other white blood cell that destroys cancer cells. And this is a good thing because some sometimes the cancer cells or infected cells lose the ability to create the MHC class 1 or the MHC class 2 complex, and that means it no longer contains these complexes on their membrane, and that makes it virtually invisible to all the cells of our body, all the white blood cells that require the MHC class 1 or class 2 to actually detect those cells. So the special thing about these natural killer cells is that they can bind and destroy these cancer cells regardless of whether or not they have the MHC complex on the membrane. So once again, natural killer cells are lymphocytes that are part of our innate immunity. Now, the majority of the white blood cells of our immune system can only recognize an abnormal cell, such as a cancer cell, if it contains an antigen that is bound onto the MHC, the major histocompatibility complex, membrane protein. However, natural killer cells are the exception. They are unique because they do not require the presence of the major histocompatibility complex on the membrane of that particular abnormal cell. Interestingly, some infected or cancer cells sometimes are missing the MHC entirely from their membrane, which makes them virtually invisible to all the other white blood cells of our body except these natural killer cells. So let's take a look at the following diagram and examine exactly what the mechanism is by which the natural killer cell detects as well as destroys our cancer cells. So when cells display little or no MHC proteins on their membrane, then that triggers the natural killer cell to basically approach and bind onto that cancer cell. So in this particular case, we have a normal cell, and a normal cell that contains a normal antigen, a normal cell antigen shown in green, as well as the MHC class 1 complex shown in brown. But this cancer cell loses the proper sequence of nucleotides to create the proper protein known as the MHC class 1. So we have none of these MHC class 1 proteins found on the membrane, and that is what triggers the natural killer cell to approach this cancer cell, bind to it, and begin releasing these granules that contain digestive enzymes. And these digestive enzymes poke holes inside the membrane of the cancer cell, and that lyses and destroys the cell. And then a macrophage can swim by and pick up that remaining debris that essentially came from that lysed cell. Now, what about cytotoxic T cells, also known as killer T cells? So we said that natural killer cells are part of the innate immunity because they destroy in a non-specific way. They are not looking for any specific cell, they're looking for any abnormal cell, including cancer cells and infected cells. On the other hand, cytotoxic T cells are part of the adaptive immunity, and that's because they are any specific cell, they're looking for any immunity because they destroy in a non-specific remaining debris that essentially came from that lysed cell. Now, what about cytotoxic T cells, also known as killer T cells? So we said that natural killer cells are part of the innate immunity because they destroy in a non-specific way. They are not looking for any specific cell, they're looking for any abnormal cell, including cancer cells and infected cells. On the other hand, cytotoxic T cells are part of the adaptive immunity, and that's because they are looking for specific infected cells, which also include cancer cells, that contain specific foreign antigens on the membrane. So these cytotoxic T cells actually need the MHC class 1 to bind onto that infected or abnormal cell. So these cytotoxic T cells contain special T cell receptors along with a CD8 glycoprotein that is needed to bind to the MHC class 1 complex. So let's take a look at the following diagram to see what the mechanism is by which the cytotoxic T cell actually kills off and finds and kills off that cancer cell. So we have normal cells in our body that display the normal self-antigen shown in green on that MHC class 1 complex. And we have this cancer cell that still has the...
My bad. Thinking it'd be helpful, but I guess not. My bad, guys.
MHC class 1 complex. Remember, display the normal self-antigen. Take a look at the following diagram to see what the mechanism is by which the cytotoxic T cell actually kills off and finds and kills off that cancer cell. So we have no complex. So let's take a look at the following diagram to see what the mechanism is by which the cytotoxic T cell actually kills off and finds and kills off that cancer cell. So we have normal cells of our body that display the normal self-antigen shown in green on that MHC class 1 complex. And we have this cancer cell that still has the MHC class 1 complex. Remember, not all cancer cells actually destroy the MHC class 1 complex. Some of them still actually have them. And in this case, the cancer cell will now display a pathogenic, a foreign antigen on that MHC class 1 complex. And now this is when the cytotoxic T cell comes into play. It has the CD8 glycoprotein and the complementary T cell receptor that can basically bind onto this particular specific antigen and onto the MHC class 1 complex. And once they are bound, the cytotoxic T cell basically does the same exact thing that the natural killer cell does. It releases these digestive proteins that poke holes in the membrane and lyse and destroy that cell. So these are the two different types of white blood cells that make up the immunosurveillance system of our body. So this is the system by which these white blood cells are continually trying to find these abnormal cells of our body, such as cancer cells, as well as our infected cells that might be cells are continually trying to find these abnormal cells of our body, such as cancer cells, as well as our infected cells that might be infected by some type of outside pathogen. So the major difference between our natural killer cell and our cytotoxic T cell is the cytotoxic T cell only binds to the MHC class 1 complex, while our natural killer cell can bind to a cell that doesn't have that MHC class 1 complex. And that's very important because sometimes these infected or cancer cells lose the ability to create these complexes that hold the antigens, and so we won't find any of these complexes on our membrane, and they will become virtually invisible to all cells of our body, including these cytotoxic T cells. But the exception is the natural killer cell. These will be able to locate these cancer cells, these abnormal cells, and release these digestive enzymes that lyse and destroy those cancer cells. Class 1 complex, while our natural killer cell can bind to a cell that doesn't have that MHC class 1 complex. And that's very important because sometimes these infected or cancer cells lose the ability to create these complexes that hold the antigens, and so we won't find any of these complexes on our membrane, and they will become virtually invisible to all cells of our body, including these cytotoxic T cells. But the exception is the natural killer cell. These will be able to locate these cancer cells, these abnormal cells, and release these digestive enzymes that lyse and destroy those cancer cells. The lymphatic system is a network of specialized vessels that plays two important roles. Firstly, it protects our body from different pathogenic infections and filters our blood from pathogenic agents that can essentially cause harm to our body and the cells of our body. And secondly, the lymphatic system also maintains fluid homeostasis. Specifically, what it does is it prevents the buildup of fluid from taking place inside the tissues of our body. Now, to see exactly what we mean by that, let's take a look at the following diagram. This diagram describes the cells of some particular tissue, and these cells are shown in brown. It also describes the capillary system that is found within our tissue. And wherever we have a capillary system, we also have a lymph system. And the vessel of the lymph system is shown in green. So basically, we have the arterial, the blood vessel, shown in red, that brings the oxygenated and the nutrient-filled blood to the capillary of our blood vessel system. And within the capillary, we have exchange between nutrients and wastes taking place. And then the venule is the blood vessel that carries the deoxygenated blood that contains the waste products back to the heart. So let's recall how the exchange of nutrients and waste products takes place within our capillary. So if we examine the arterial side of the capillary, on the arterial side, we have a higher hydrostatic pressure than osmotic pressure. And that's exactly why that hydrostatic pressure is able to force the blood plasma that contains the nutrients and our oxygen from the capillary and into the surrounding tissue space. And the surrounding tissue space is known as the interstitial tissue. So what happens is when the blood plasma leaves the capillaries and enters this cell tissue area, that brings the nutrients such as glucose and fats and amino acids and oxygen to the cells of that surrounding tissue. And at the same time, as that fluid travels along the tissue space, along the interstitial tissue, it picks up those waste products that are secreted by the cells, such as carbon dioxide and ammonia. And once the fluid is on the venule side, on this side of the capillary, because the osmotic pressure is now greater than the hydrostatic pressure, the blood rushes back into the capillary of our body. And now the deoxygenated blood that contains the waste products travels along the venule, then into the veins, and finally into the heart of our body. Now, it turns out that osmotic pressure on the venule side is not that much higher than the hydrostatic pressure. And what that means is not all of that blood plasma that left the capillary actually returns back into that capillary on the venule side. In fact, about 10% of that fluid that left the capillary and entered our tissue will remain in that interstitial space, in the space surrounding our capillary. The question is, what happens to this 10% fluid? If this 10% fluid is not removed in any way, there will be a buildup of pressure as a result of the buildup of fluid inside that tissue, and that will lead to swelling, the process of edema, and that can lead to very serious medical conditions and medical complications. So to prevent this from happening, what our body does, and specifically what the lymphatic system does, is it drains and removes that fluid into the system of vessels. We call lymph vessels or lymphatic vessels. These lymph vessels essentially connect with larger lymph vessels known as lymph veins, and along the lymph vessels, we have these regions known as lymph nodes, and we'll talk about them in just a moment. And these lymph nodes essentially filter our lymph from different types of pathogenic agents, and eventually that lymph is returned back into our blood system via specific types of veins, as we'll see in just a moment. And the reason we want to return our lymph back into our blood is because we want to ensure that the same volume of blood remains in our cardiovascular system. So once again, the question is, what happens to the 10% fluid that remains in the tissue space? If it remains in the tissue space, it will lead to a continual buildup, the process of swelling, the process of edema. And to prevent this from happening, our body uses lymph vessels, shown in green, to drain this fluid out of the interstitial space. Now, the fluid, which is now known as lymph, travels along these venules, along these vessels, and eventually connects with these larger vessels we call lymph veins. And eventually the lymph veins reconnect with the blood vessels, and the fluid is returned back into our blood circulation through the thoracic duct and the right lymphatic duct that both connects with special types of veins. So to see what we mean, let's take a look at the following diagram. So we have two important types of ducts, the thoracic duct as well as our right lymphatic duct. The thoracic duct essentially collects the lymph from the lower right part of the body, from the GI system, and from the left side of the body, the entire left side of the body, and it connects with our circulation system via the left subclavian vein. So this is the brachiocephalic vein, this is the right subclavian vein, and this is the left subclavian vein. Remember, the subclavian veins carries the deoxygenated blood from the arm portion and into the vena cava, which brings that blood to the right atrium of our body. And along the right subclavian vein, we have a connection between that vein and the right lymphatic duct. while along this left subclavian vein, we have a connection between this thoracic duct and the left subclavian vein. So the lymph, once we actually filter that lymph, it returns back into the vein system of our body and back into the cardiovascular system. So we see that the right lymphatic duct collects the lymph from the right side of the head, the neck, and the chest, and empties into the right subclavian vein. Now, previously, we mentioned that one of the other functions of the lymphatic system is to filter our blood, to filter our lymph that travels along these lymph vessels. And this filtering process takes place in lymph nodes. So along many parts of our lymphatic system are small masses of tissue called lymph nodes. So this is one lymph node, a second lymph node, a third lymph node, and we have many of these lymph nodes along different regions of our body. Now, within these lymph nodes, we have cavities, we have sinuses, and within these cavities, we have specialized types of leukocytes, white blood cells. Now, when dendritic cells found in the tissue pick up pathogenic antigens, they carry these pathogenic antigens into our lymph nodes. And inside the lymph nodes, we have plasma cells that produce antibodies against these antigens. And these antibodies basically leave the lymph nodes along the other side, and eventually they are dumped into our vein system. And that's how antibodies end up in the cardiovascular system, in the blood vessels of our body. Now, within these lymph nodes, we also have other white blood cells, such as macrophages, that can engulf any type of pathogenic agent that might be present inside our lymph system. And in this manner, our lymphatic system not only drains our tissue and prevents the buildup of fluid inside the tissue, but it also filters our blood. It basically eats up and digests different types of pathogens that are found inside our blood, inside these specialized masses of tissue we call lymph nodes. Now, the final portion that I'd like to focus on is how that fluid actually gets into these lymph vessels in the first place and how the lymph travels along our lymph vessel. So let's take a look at the following diagram. So this is a small portion of our lymph vessel. Now, notice along the lymph vessel, we have these endothelial cells. So the walls of the lymph vessels consist of endothelial cells that overlap slightly. And at the portions where they overlap, these overlapping portions act as one-way doors. And when there is a fluid buildup inside the tissue, that fluid pressure pushes on these overlapping sections, these one-way doors, and that opens these endothelial cells and allows fluid to actually flow into the endothelial cells. So for example, let's imagine that we have a buildup of pressure here, and so the fluid pushes against the overlapping portion, and the fluid moves into this cavity, this region of our lymph vessel. Now, as the fluid builds up in the lymph vessel, it pushes back onto this side, the other side of our endothelial cell. And because these endothelial cells open only one way and not the other way, when our hydrostatic pressure pushes on our cells this way, the fluid cannot escape back because these overlapping regions between the cells only open this way and not the other way. And we also have a system of one-way valves found along the lymph vessel. So that means these valves also open one way and not the other way. So when there's a buildup of pressure here, it closes these overlapping regions so that fluid cannot exit that lymph node. It pushes against a valve that opens up, and that allows the movement of lymph along our vessel as shown in the following diagram. And if there is a decrease in pressure and it basically wants to move back, it cannot move back because when the fluid tries to move back, it forces these valves to actually close, and that means we do not have a backflow of lymph inside our lymph system in the same way that we do not have a backflow inside our veins. Remember, the veins also contain this valve system that prevents the movement of our lymph back down that lymph vessel. So the walls of the lymph vessels consist of endothelial cells that overlap slightly. When there is a buildup of fluid in the interstitial tissue, in the tissue space, that creates fluid pressure that pushes on the cells. This opens up those overlapping regions, forces the fluid into that lymph vessel. Now, inside the lymph vessel, we have a system of one-way valves. These valves, as well as the overlapping portion of the endothelial cells, open only in one direction and not the other. And this keeps the lymph inside that lymph vessel, and it keeps it moving along one direction. Now, in the same way that our veins are situated next to our skeletal muscle, and the contraction of our skeletal muscle promotes the movement of blood inside the veins, in the same exact way, the contraction of skeletal muscle pushes against our lymph vessels, and that promotes the movement of lymph inside our lymphatic system.
Wow. That's good. Pakistan. Nice. Very cool. All right, let's Yusuf Hassan. Let's see. I mean, Enrique is pretty good. Yusuf Hassan. Oh, he doesn't have much videos. Did I get it right? Yusuf Hassan. Is this the right person? Is this him? My camera? (2021) videos? Which one's that one? Yeah, it is. Jack Weston biology. Oh, the old one. There's not that many videos. Okay. Are they like
Let's chat.
Oh, they're quite long.
So I'm gonna tell a little story about this lecture hall specifically. So obviously I went from college, and the first class I was ever in when I came from college was an intro bio course. And that intro bio course was taught by Professor McEntee. I don't know if she's still here, but she's a fantastic
Wow, that's pretty
professor.
good.
I hope that you guys all have the opportunity to cross paths with her. But she taught in this lecture hall. And that was my first class ever. And I was seated right there in like the third to last row.
Okay, well, I'm doing bio, so let's also do biochem. Yes, sir. Anyways, this is pretty good. Yeah, his videos are quite long. That's fine. I mean, I could go through them though. Sure. I can include that in my review. His biochem is especially good. Yeah. Okay. Understood everything. Nice. Okay. Yeah. I can watch him. Well, we might as well, right? Okay. So, I'll just keep it here. So, biochem. Then I want to do. Well, I want to do 1 week of PS for me, and then Gen Chem. Okay, I actually want to do physics last. Let's read the books. And the books. Should be going. Just videos. Oh, okay. Nice. Yeah. Well, it's been some time for me, so. But I'm almost done with the Khan Academy videos, so I plan to just finish those this week. This week I also have the book too. Yeah, I have the book. That's what I was gonna go over this week. Yeah. I guess I can just bring it up for now. Oh, jeez. Yeah. I could leave it off to the side. Yeah, I want to do biology first. Yeah, okay. Thank you. I appreciate it. I will go through the videos. Don't worry. I will make sure to go through his videos too. I mean, we're almost done with biology, to be honest. And then I want to take full length, first full length, but just focus on the baby section first. So I still have some time. Actually, I have to do Anki right now. I have to do quite a lot here because I kind of fell behind. So I don't have you world though. He doesn't have physics. Yeah, that's a shame. But I have the books, and some people say Jack Weston. So uh, it's one thing I should get. Okay. For the test, I'm prepping for first year medical school finals coming up in about 2 months. Kind of dying. Cars. Bootcamp cars is free for 10 months. What is bootcamp cars? What is that? What is bootcamp cars? Is that bootcamp.com? Oh, okay. All right. Thank you. Thank you so much. I appreciate that. Thank you so much. Yeah. We'll just leave it down here, actually. Okay. We have quite a lot to retweet today, to be honest. Oh, okay. Yeah, let me write it down. Okay, actually, I just wrote it down. Is that from the website MCAT Bootcamp? Is it this one? Is it that one? Oh, you might not be able to see the link. I'm not sure. Okay, cool. Thank you. Oh, signal. Oh, hemoglobin. Hemoglobin. Got off hemoglobin. Yeah, thank you so much. Thank you. Yeah, thank you. That's cool. That's nice. Thank you. Appreciate it. Thank you. All right. Yeah. Analytic material page. Zoom into the slides. There is a way to do something. Lysogenic. Hmm. Yeah. Oh, that's a GABA. Well, enzymes. Yeah, that's a good one. Okay. Breakdown. Breakdown by enzymes. Reactive receptor diffusion. Oh yeah, reuptake. I wasn't even thinking about that. Breakdown by enzymes. Yeah. Oh, and diffusion. Okay, yeah, that's fine. Africans? Yeah. Sensor for touch, light, sound. Touch, light, sound. Well, anything sensory. Integration. %. That's the first. I'm not sure what the number is. P50. P50. Oh, okay. I don't know. It's okay. Okay. P50. 50% hemoglobin has an oxygen power. That's the right shift. Okay. More positive. Good nation. Pathogens clump together. So that's pathogens clump together into insoluble complexes. Preference. Transports proteins. Modifies and distributes proteins in the trans. Functions to decrease calcium in the blood. That's increase actually. Oh no, yeah. That's decreased calcium in the blood. Huh. Is that true? Okay. Hollow tube that connects to the layers. No. Layers of phalanx. And no layer to the frontal ones. The layers to the bronchi of the lungs. Silated epithelium collects the green of the trachea and the lung. When O2 concentration is 50%. I need to pee. I need to pee. Hold on. And we'll just do 2 hours. I'll set the clock when I get back. Yeah. All right, guys. I think I'm actually gonna call it here. Let's take some NyQuil, take a shower, and then let's go to bed. I'll wake up early tomorrow. Thanks for watching, everyone. I'll see you guys tomorrow. Peace.